We are talking about a virion diameter of just 28 to 30 nanometers.
Right, and numbers like nanometer can be really hard to visualize. So to give you a sense of scale, if a human red blood cell, which is already microscopic.
All right, you can't see it.
Exactly. If that red blood cell were the size of a massive football stadium, a picornavirus would be roughly the size of a marble, just sitting right there on the 50-yard line.
That is a perfect analogy. They are on the very edge of what can physically exist as a biological entity. I mean, if they were much smaller, they simply wouldn't have enough physical room to pack the genetic constructions they need to replicate.
And yet, and here's the paradox that really jumped out at me. You have these microscopic specks, right? This marble in the stadium. Yet when you look at what they do to the human experience, it's a staggering range of misery.
It really is a massive spectrum.
On one end of the spectrum, you have the common cold. We all get it. It's annoying. You know, you watch some bad TV on the couch for 2 days and you're fine.
Right. It's a nuisance.
But on the other end of the spectrum, from the exact same family of viruses, you have life-altering paralysis, heart disease, fatal meningitis, and diseases that can literally decimate livestock economies.
That is the Picornavirus paradox, if you will.
Yeah.
How does something so physically small and genetically simple, we're talking about a very compact genome here, barely a pamphlet compared to the encyclopedias of other organisms, how does it wreak such diverse havoc? That is really the mission of our deep dive today.
Right. We're going to unpack the entire Chapter 36 source material to understand the mechanics, the history, and really the pathology of this whole viral family.
And it is quite a lineup of characters we have to get through today.
Oh, yeah. We've got the Enteroviruses, which I always think of as the gut group.
That's a good way to remember it.
And that includes the heavy hitters, right? Polio, Coxsackie, and Echo.
Correct. They are called enteroviruses because they are inhabitants of the human alimentary tract. They primarily replicate in the intestines, which is a key distinction we'll get into later. They are tough, rugged survivors.
Then we have the Rhinoviruses, which is the nose group, the architects of the common cold.
And we can't forget the outliers, like the Aftovirus genus.
Right, which causes foot and mouth disease. It's a huge issue for livestock, even if it doesn't affect humans in the same way.
Exactly.
And before you check out thinking, oh, the common cold, I know that, or polio is solved, why should I care?
Yeah.
Let me just say, the stakes here are deceptively high. The biology we are looking at reveals some incredibly sophisticated survival mechanisms.
Absolutely mind-blowing mechanism.
We are going to talk about literal canyons on the virus surface that hide from your antibodies.
That is a fascinating piece of evolutionary engineering. It's essentially a microscopic fortress design.
It's crazy, and we're going to look at why global eradication is so difficult. So let's get into the blueprint. Let's talk about what these things actually look like. You mentioned 28 to 30 nanometers. What shape are we dealing with? Is it just a blob?
Far from it. It's a piece of precise geometry. Imagine a tiny soccer ball-like shape. That's called icosahedral symmetry.
Okay, icosahedral.
Yeah, and it's composed of 60 subunits. But it's not just a smooth ball. This protein shell, or capsid as it's called, is incredibly complex.
The source material breaks this down into specific proteins, VP1, VP2, VP3, and VP4. Now, usually protein names make my eyes completely glaze over.
I get that.
But the arrangement here is actually really important for how the virus works, right?
It is crucial. So picture the surface of that soccer ball we just talked about. VP1, VP2, and VP3 are the proteins that make up the outer shell, and they fold in a very specific way. The text calls it a beta barrel.
A beta barrel. That sounds structural.
It is highly structural. Essentially, the peptide backbone loops back on itself to form a barrel of 8 strands, all held together by hydrogen bonds. It creates a very sturdy armor. This isn't a fragile little bubble. It's a reinforced tank.
Okay, so you have this sturdy barrel structure on the outside made of 1, 2, and 3. Where's VP4?
VP4 is tucked inside. It's an internal protein. It's effectively anchoring the RNA core on the inside. But that surface arrangement of the other 3 creates something that I think is the most brilliant part of this virus's design, the canyon.
Yes, the canyon hypothesis. I love this concept. It sounds like a sci-fi landscape or something. Describe what this actually looks like on the virus.
Okay, so imagine the surface of the virus again. Around each of the pentameric vertices, those are the points of the icosahedron, the corners of the soccer ball, there is a deep cleft or a depression. We call it a canyon.
Like a moat around a castle.
A moat is a pretty good analogy, but think of it more like a really narrow trench. Now, at the very bottom of this trench on the floor of the canyon is the receptor binding site.
Okay.
That is the key the virus uses to unlock the door to the human cell.
So wait, The critical part of the virus, the part that must touch the cell to infect it, is buried at the bottom of a hole.
Yes.
Why? What is the strategic advantage there?
It is a brilliant defense against your immune system. Your body produces antibodies to fight viruses, right?
Right.
These antibody molecules are Y-shaped, and they are relatively bulky. The source text explains that the canyon is roughly 12 to 30 angstroms wide.
Which is too narrow for the antibody to fit.
Exactly. The antibody is physically too fat to reach the bottom of the canyon. It can bind to the rim of the canyon or the top of the plateau, but that doesn't stop the virus from working.
Why not? If the antibody grabs the rim, isn't the virus stuck?
Not necessarily, because here is the genius part. The virus can mutate those surface loops on the rim. It can change its disguise constantly. So your immune system makes antibodies for the rim, but by the time they are ready and circulating, the virus has mutated and the rim has changed shape.
Oh, wow. But it cannot change the floor of the canyon.
Precisely. If it changes the floor, the key no longer fits the lock and it can't infect the cell anymore.
Right.
So it hides the one part it cannot afford to change at the bottom of a hole where your defenses physically can't reach it.
That is diabolical.
Isn't it?
It completely preserves the critical function while escaping immune surveillance. It's this perfect evolutionary balance between protection and function.
It really is. It's a microscopic standoff that has lasted for millions of years.
And inside this highly defended fortress is the genetic core. We said earlier it's RNA.
Yes, a single strand of positive sense RNA. And attached to the end of it is a very small viral-coded protein called VPG.
VPG.
Right. That VPG is covalently bound to the 5' end of the RNA, and it is absolutely essential for the virus to replicate. It acts almost like a primer to get the copying process started once it's inside.
Now, I want to distinguish between the two main groups we're talking about today, enteroviruses and rhinoviruses. Structurally, they look similar, right? They both have this canyon, this beta barrel armor.
Yes, the basic blueprint is the same.
But they have very different tolerances for their environment.
This is a key diagnostic difference, and it dictates exactly where they live in your body. Enteroviruses are stable at acid pH. We're talking a pH of 3.0 to 5.0.
Which makes sense, if you think about it. If they are entero-gut viruses, they have to survive stomach acid.
Correct. To reach the intestines, you have to pass through the stomach, which is essentially a vat. It would be digested like a piece of steak, but it has evolved to treat stomach acid like a warm bath.
Wow. And on the other hand, rhinoviruses are acid labile.
Yes. Labile, meaning fragile or easily broken down. They are completely destroyed by acid. That is exactly why rhinoviruses don't infect your gut.
They stay in the respiratory tract.
They live in the nose and throat, where the pH is neutral. If you somehow swallowed a rhinovirus, your stomach acid would obliterate it instantly. That's why you don't get a stomach flu from a common cold virus.
Makes so much sense. There's also a density difference mentioned in the text involving cesium chloride.
Yes, that's a bit of laboratory trivia, but it's how scientists separate them. They use a high-speed centrifuge with cesium chloride. Enteroviruses have a buoyant density of about 1.34 grams per milliliter.
Okay.
Which is slightly heavier than rhinoviruses, which sit at 1.4 grams per milliliter.
Wait, 1.4 is heavier than 1.34.
Excuse me, you are absolutely right. The rhinoviruses are the heavier ones at 1.4. Enteroviruses are lighter at 1.34. It's a subtle physical property, but it helps researchers identify what they are looking at in a sample.
Good catch. So we have the blueprint. A tiny armored tank with a hidden key in a canyon, carrying a very specific RNA payload.
Yes.
Now let's talk about the heist. How does this thing actually break into a human cell and hijack it?
Well, it starts with that canyon we just discussed. The virus floats up to a cell. The canyon floor locks onto a specific receptor on the cell's surface. And interestingly, different members of the Picornavirus family use different doors to get in.
Right. The source lists a few specific ones. Poliovirus uses something called PVR or CD155.
Right, which is part of the immunoglobulin superfamily. It's just a normal protein your cells naturally display. Rhinoviruses, the common cold, mostly use ICAM1.
ICAM1, what is that normally used for?
It's an adhesion molecule. Your cells use it to stick to each other during normal tissue function.
So the virus just opportunistically hijacks that sticky handhold.
Exactly. And echoviruses use integrins. They find a handle and they grab it.
So the virus floats up, the key in the canyon locks into the receptor.
Yeah.
And then what happens? Does it just sort of merge with the cell membrane?
No, it's much more aggressive than that. Binding actually causes a massive conformational change in the virus structure. Remember VP4, that internal protein we mentioned earlier?
The one tucked inside the shell anchoring the RNA?
Right. It gets ejected.
Ejected.
Yes. The virus essentially cracks open. It creates a pore right in the cell membrane and injects its RNA directly into the cell cytoplasm.
It effectively acts like a syringe.
In a way, yes. It leaves the empty protein shell behind on the outside, or it gets broken down, but the genetic material is now inside your cell. And this is where the takeover becomes, and I don't use this word lightly, ruthless.
The text mentions something about the virus completely bypassing the normal rules of the cell.
It does. Let's look at how your cells normally work. Normal human messenger RNA has a cap structure at the beginning of the strand. It's like a VIP ticket or a barcode that tells the ribosome, which is the cell's protein factory, hey, start printing here.
Okay, so the cell looks for the cap.
Right. But picornaviruses don't have that cap. They are uncapped RNA.
So how do they get printed? If they don't have the VIP ticket, shouldn't the ribosome just ignore them?
You would think so. But they have evolved a structure called an IRES, I-R-E-S. It stands for Internal Ribosome Entry Site. It allows the virus to cut the line, essentially. It grabs the ribosome directly and says, start reading here, forget the cap.
That's incredibly sneaky. But the virus isn't content with just cutting in line, is it? It does something worse.
Much worse. It wants to be the only one in line. The source describes how the virus often produces a protease, which is an enzyme that cuts other proteins.
Okay.
And this viral protease actively hunts down a specific cell factor called EIF4.
EIF4. That's a host translation factor, right?
Yes. And it is absolutely required for the cell's own capped mRNAs to be read. By chopping up EIF4, the virus effectively destroys the cell's ability to read its own instructions.
Wow. So it walks into the factory, breaks the machine that reads the factory's own normal blueprints, and then installs a bypass that only works for the virus's blueprints.
Exactly. It forces the cell to devote all its energy, all its raw materials to making virus parts. Cell maintenance stops, cellular defense stops. The cell becomes a zombie factory solely for the virus.
That is terrifyingly efficient. And the way it actually makes those parts once it takes over is also unique. It uses what the text calls a polyprotein strategy.
This was really interesting to read about. Usually in human cells, genes are read one by one to make individual proteins, but the picornavirus translates its entire genome into one giant continuous protein strand, a polyprotein.
I was trying to visualize this. It's like, imagine printing a car, but instead of making a tire and then a door and then an engine separately, you print one long, shapeless blob of plastic and metal that contains all those parts fused together in a line.
That's a great visual. And then the snake cuts itself.
The snake cuts itself.
Right. The virus uses its own proteases, which are actually embedded right there in that giant strand, to rapidly fold and cleave the strand into the functional parts. It chops itself up into the capsid proteins, the RNA polymerase, the proteases.
It's incredible. Just pure efficiency.
It is. And once it has those separated tools, it starts copying itself furiously. The RNA polymerase makes negative strands from the positive ones and then uses those negative strands as templates to pump out huge numbers of new positive strands.
And some of those new positive strands go back to the factory floor to make more protein, while others get packaged into new viral shells.
Precisely. And then comes the exit strategy, which is not subtle. It's lysis.
It blows up the cell.
Yeah. Maturation involves a final cleavage step where a precursor called VP0 snaps into VP2 and VP4, creating the fully infectious virus particle. Then the cell membrane disintegrates, releasing thousands of new virus particles to infect all the neighboring cells.
And how long does this whole hijacking take?
The whole cycle takes just 5 to 10 hours.
5 to 10 hours. That is so fast. You can see how an infection spreads so quickly within the body. You know, a sore throat goes from maybe I'm getting sick to I literally can't swallow overnight because of this exponential timeline.
It's a race. And because they shut down the host cell's normal maintenance immediately, the damage is incredibly rapid.
Let's pivot to talk about the actual diseases this family causes. We have to start with the heavy hitter, polio.
Poliomyelitis. It is, without a doubt, the most serious disease caused by enteroviruses. For most of human history, it was an endemic companion, just always there in the background. But in the 20th century, it became an absolute terror.
The text traces the exact path of the virus through the body, which is super helpful. It starts in the mouth, obviously.
Right. It's fecal-oral transmission, usually. It enters the mouth, and it multiplies in the oropharynx, the back of the throat, and the small intestine. Specifically, it targets the Peyer's patches, which are immune tissues lining the gut.
So it's breeding in the gut. But then it moves.
From the gut, it moves into the local lymph nodes, and then it spills over into the blood. That stage is called viremia.
So at this point, you have virus circulating in your blood. But polio is famous for paralysis. How does it get from the bloodstream into the nerves?
That is the critical, devastating step. It manages to cross the blood-brain barrier into the central nervous system, the CNS, and it begins to spread along nerve fibers. Its specific target is the anterior horn cells of the spinal cord.
Anterior horn cells.
Yeah.
Those are motor neurons, right? The ones that control movement?
Yes. They are the physical wires that send signals from your brain to your muscles. When the virus multiplies inside these cells using that lysis process we talked about, it destroys them. And when the neuron dies, the connection is permanently cut. The muscle it connects to can no longer receive signals.
And that causes flaccid paralysis.
Flaccid, meaning the muscle goes completely limp. This is distinct from spastic paralysis, where muscles get rigid and tight. In polio, the muscle becomes loose, useless, and eventually it atrophies because it's not getting any electrical stimulation anymore.
But here is the thing that always surprises people, and the source text highlights this very clearly. The vast, vast majority of polio infections do not end in paralysis.
That is a crucial point to understand the epidemiology of the disease. We tend to think of the word polio and immediately imagine the iron lung wards or kids in leg braces. But the source material notes that about 99% of infections are either completely asymptomatic or result in just a minor illness.
Like a mild fever.
Exactly. Fever, malaise, maybe some drowsiness or a sore throat.
So you could have polio and literally just think you had a mild flu.
Exactly. It's a classic tip of the iceberg disease. The visible paralysis is just the tiny tip. Below the surface, there's a huge base of mild infections spreading the virus.
And then there's a slightly smaller group that gets what's called aseptic meningitis.
Right. That involves stiffness in the neck and back, severe headache. It's scary, but they generally recover fully. Paralytic poliomyelitis, the classic dreaded presentation, actually occurs in only about 1% of total infections.
But that 1% is devastating. And the text mentions a specific type called bulbar polio.
That's when the virus attacks higher up into the brainstem, affecting the cranial nerves. This is particularly lethal because it can paralyze the respiratory muscles. Your diaphragm stops working.
And that's where the iron lung came in.
Yes. There were negative pressure ventilators. They literally breathed for people who couldn't expand their own chests because the nerves were dead.
There's also a section in the text on post-polio syndrome, which I found really tragic. It's muscle atrophy returning decades later.
It is incredibly tragic. You have people who survived paralytic polio in the 1950s, worked hard, recovered some function, lived their lives, and then 30 or 40 years later, they start getting weak again, losing function they had fought to regain.
Is the virus back? Is it hiding in them all that time?
No, and that's the important distinction to make. It's not a reactivation of the virus like shingles. The text explains it as essentially wear and tear.
Wear and tear on the nerves.
Right. The idea is that when the original neurons died during the infection, the surviving neurons sprouted new endings to try and take over the work of the dead ones. They were doing double or triple duty for decades.
Oh, I see.
But over time, those overworked, massively overextended neurons essentially burn out and die prematurely.
So it's a long-term physiological consequence of the initial damage, not a new active infection.
Correct. It highlights that the damage from polio is truly lifelong, even if you supposedly recover.
I want to touch on the epidemiology of polio because there is a really counterintuitive point in the text about hygiene. It says epidemics actually increased when hygiene improved. Now, that sounds totally backward. We are universally taught that clean water stops disease.
It does seem backward. It's called the paradox of hygiene, and it makes perfect sense if you look at the immunology of a population over time. In developing countries with poor sanitation, or if you look back at the 19th century globally, poliovirus was everywhere. It was in the water, the soil. So infants almost inevitably get infected very, very early in life, often while they are still nursing and still have maternal antibodies circulating in their blood from their mothers.
So the mom's antibodies protect the baby from the paralysis, while the baby gets that mild gut infection and builds its own lifetime immunity.
Exactly. It acts almost like a natural, relatively safe vaccination program. The entire population becomes immune as infants without seeing much paralysis at all. But as sanitation dramatically improved in the 20th century, especially in cooler climates.
The fecal-oral transmission chain was broken?
Right. Flush toilets, clean municipal water. Infants stopped getting exposed to the virus early in life.
So they grew up completely without immunity.
Yes. They became a huge population of susceptible older children and teenagers, immunological virgins to the virus. So when the virus finally did arrive in a community, maybe imported by a traveler or just through a gap in sanitation, it hit older children who didn't have that maternal protection anymore.
And it spread like wildfire through the schools and pools, causing severe paralysis.
Exactly. So cleaner water and better sewage actually set the stage for the massive, terrifying polio epidemics of the mid-20th century.
That is mind-blowing.
It's a classic example of how disease patterns shift dramatically with societal changes. We shifted from an endemic, mild pattern to an epidemic, paralytic one.
But then, of course, came the heroes of the story. The vaccines. We have two main players detailed here, Salk and Sabin. The text breaks down the difference between them perfectly, and it's super important.
It is. So Jonas Salk developed the killed or inactivated polio vaccine, IPV. You inject it into the muscle. Because it goes into the tissue and blood, it heavily stimulates the production of IgG antibodies in the bloodstream.
Okay, so if the wild virus gets in your mouth, goes to your gut, and tries to cross into the blood, the IgG antibodies are there waiting to stop it from reaching the nerves.
Correct. No paralysis. It is incredibly effective at preventing the disease. But, and this is a big but for public health, because it's injected, it doesn't induce strong localized immunity in the lining of the gut itself.
So the wild virus can still live and replicate in your intestines.
Yes. You, the vaccinated person, are perfectly protected from getting sick. But wild virus can still replicate quietly in your gut, and you can shed it in your stool for weeks, potentially infecting others who aren't vaccinated.
So it protects the individual, but it's less effective at stopping the spread of the virus in a community.
Right, which is exactly where Albert Sabin's vaccine comes in. That's the oral one, OPV, the one given on a sugar cube or as drops in the mouth.
And that one is a live virus.
A live attenuated virus, meaning it's weakened so it shouldn't cause disease. Because you swallow it, it mimics the natural route of infection. It sets up shop in the gut and induces a strong local response of IgA antibodies right there in the intestinal lining.
So it fights the virus at the front door.
Exactly. This actually stops wild virus from replicating in the intestines at all.
Meaning Sabin stops transmission.
Yes, it provides excellent herd immunity. If you give it to enough people in a village, the wild virus literally has nowhere to replicate and it dies out. It essentially puts a massive do not enter sign on everyone's gut.
But it has a risk.
It does. Because it is a live virus, it replicates. And every single time a virus replicates, it can mutate. In very, very rare cases, we're talking about one case for roughly every 2 to 3 million doses given, the weakened virus can mutate back to its dangerous form.
It reverts.
Yes, it reverts to neurovirulence, and it causes paralysis in the person who took the vaccine or a close contact.
Vaccine-associated paralytic poliomyelitis, VAPP.
It's a cruel numbers game. When wild polio was rampant and paralyzing thousands of kids a year, taking that 1 in a million risk was absolutely worth it to stop the outbreak. But once wild polio was functionally eliminated from countries like the U.S.,
The only people getting polio were getting it from the vaccine.
exactly, the risk calculus flipped. So in 2000, the United States switched entirely back to the Salk vaccine, the IPV. We prioritized individual safety over the stronger gut immunity because the threat of wild virus circulating was basically zero.
But in places where polio is still endemic, like parts of Afghanistan or Pakistan, they still use the Sabin drops.
They have to. They are actively trying to stop community transmission. You need that gut immunity to break the chain. The global eradication effort relies heavily on the oral vaccine to stamp out outbreaks, and then regions transition to the injected vaccine once it's clear.
Fascinating strategy.
Yeah.
Okay, let's move away from polio to its diverse cousins, the Coxsackie viruses.
Named after the small town of Coxsackie, New York, where they were first isolated from patient stool samples back in the 1940s.
The text divides them into two main groups, Group A and Group B. And the list of symptoms here is, well, I called it a grab bag earlier. It's incredibly diverse.
It is, and interestingly, the initial classification into A and B wasn't based on human symptoms, but on what the viruses do to newborn mice in the lab.
Oh.
Yeah. Group A causes widespread myositis, severe muscle inflammation, and flaccid paralysis in mice. Group B causes spastic paralysis and fat tissue necrosis.
But for us, the human symptoms are what we care about. Let's talk about herpangina. That sounds awful.
It is unpleasant. It's caused by Group A Coxsackie viruses. It's a severe febrile pharyngitis. You get these painful vesicles or small blisters on the back of the throat and the palate.
Ouch.
It is very common in young children. It comes on very abruptly with a high fever and a terrible sore throat.
And then there's hand, foot, and mouth disease. Now, I have kids, so I definitely know this one.
Most parents do. It's usually caused by Coxsackie A16, though sometimes Enterovirus 71. You get a vesicular rash, little red blisters on the palms of the hands, the soles of the feet, and inside the mouth.
It spreads through daycares like wildfire.
Highly contagious, but it usually resolves on its own in a week or so without crusting over, unlike chickenpox.
Okay, then we have Group B, which seems to get a bit scarier. Pleurodenia. The text notes it's also called the devil's grip.
That is a very vivid name, isn't it? It's also known as Bornholm disease. It causes sudden, stabbing, agonizing chest pain.
That sounds like a heart attack.
It frequently mimics one, bringing people to the ER, but it's actually severe inflammation of the muscles between the ribs or the lip lining the lungs. The pain is spasmodic and gets worse when you try to breathe deeply.
And speaking of the heart, Coxsackie B is a major cause of myocarditis, right?
Yes. Inflammation of the heart muscle itself, or the sac surrounding it, the pericardium. This can be very serious. It can lead to permanent heart damage, arrhythmias, or congestive heart failure. It affects both infants and healthy adults. In fact, the text notes that about 5% of all symptomatic Coxsackie infections induce some level of heart disease.
That's a significant percentage for a virus people barely think about. There is also a fascinating note in the source material about diabetes.
Yes, this is a major topic of ongoing research mentioned in the chapter. There is strong epidemiological and laboratory evidence that Group B Coxsackie viruses can attack the beta cells of the pancreas.
And those are the cells that manufacture insulin.
Right. And the theory is that this viral damage, or the immune system's response to it, triggers the development of type 1 diabetes in genetically susceptible people.
So a random viral infection in childhood might be the trigger that starts the autoimmune destruction of the pancreas.
Exactly. It completely changes how we think about the origins of chronic diseases. It highlights how these supposedly simple viruses can have incredibly complex long-term interactions with our biology. It's not just a passing cold. It could be the start of a lifelong chronic condition.
Wow, okay, moving on to the next group in the family, the echoviruses and paraechoviruses. I love the story behind the name ECHO.
It's a great acronym. ECHO. It stands for Enteric Cytopathogenic Human Orphan.
Orphan, that implies it didn't have a disease family.
Precisely. Back when cell culture techniques were getting really good, scientists were swabbing stool samples and finding all these new viruses. They saw they killed cells in a petri dish. That's the cytopathogenic part. And they lived in the gut enteric.
But they couldn't link them to a patient actually being sick?
Right. The patients were often fine, so they were considered orphans in search of a disease.
But they found it home eventually, didn't they?
They did. As surveillance got better, we realized they weren't harmless. We now know echoviruses are major causes of aseptic meningitis, various summer rashes, and infantile diarrhea. They just weren't obvious at first glance.
The text also highlights a couple of specific enteroviruses that have actually made scary headlines recently. Enterovirus 68.
EVD68. The source specifically mentions the large outbreak in 2014 in the U.S. It caused severe respiratory illness, mostly in children who already had asthma. But shockingly, a small, tragic fraction of these cases were associated with acute flaccid paralysis.
Acute flaccid paralysis. That must have been terrifying for public health officials, seeing polio-like symptoms cropping up again from a different virus.
It was a massive wake-up call. It shows that other enteroviruses can potentially mutate and fill the neurological niche left empty by polio. EVD68 is also weird because it shares some characteristics with rhinoviruses, like being acid labile, which made it confusing to classify at first.
Then there is Enterovirus 70, which causes acute hemorrhagic conjunctivitis.
Bleeding eyes.
Literal bleeding eyes.
Essentially, yes. A very sudden onset of subconjunctival hemorrhage. Your eye turns bright red. It appeared in a massive pandemic form in 1969 in Africa and Southeast Asia. Very contagious, spreads rapidly in crowded or unhygienic conditions.
And what about paraechoviruses? The text says they used to be echoviruses, right?
They were. They were formerly classified as echovirus 22 and 23. But as genetic sequencing got better, scientists realized their genome organization is significantly different from the rest. So they got their own genus. They mostly cause respiratory and gastrointestinal illness in young infants. It's just another example of how viral taxonomy is always evolving as we look closer at the code.
Okay, let's talk about the one member of this family everyone listening has definitely had, probably dozens of times, the rhinovirus. the universal nuisance.
The common cold. It is by far the most common cause of mild upper respiratory infections in humans.
We mentioned earlier that they are acid labile, so they get destroyed by the stomach and don't go to the gut. But the text says something really interesting about their temperature preferences. They like it cool.
They do. In the lab, they grow optimally at 33 degrees Celsius.
Which happens to be?
The temperature of your nasal mucosa,
Yeah.
the lining of your nose where the air is rushing in and cooling the tissue.
Which is distinctly cooler than your core internal body temp of 37 degrees.
Right. So the human nose is their perfect evolutionary incubator. They literally don't replicate, well, deep in the lungs where it's 37 degrees, which is a big reason why they usually stay confined to the upper respiratory tract. It's a very specific, elegant adaptation.
Now, here's the question everyone always asks. Why can't we cure the common cold? We can put a rover on Mars, we eradicated smallpox, we have vaccines for polio, but I still get the snivels and a sore throat twice a year.
It all comes down to diversity. Think back to polio. There are exactly 3 serotypes of wild poliovirus, type 1, 2, and 3. You put all 3 in the vaccine, your body learns them all, and you are done.
And rhinovirus.
There are over 100 known serotypes. The newer data in the text mentions there are likely more than 150. And the immunity your body builds is entirely type-specific.
So if I catch rhinovirus type 14,
You build antibodies, and you are immune to type 14, but you are still completely 100% vulnerable to type 15, type 16, type 89, and so on. There is basically no cross-protection.
and the immunity doesn't even last forever, does it?
No, the text notes that antibody levels drop off over time. It's transient immunity. So you could theoretically get reinfected with that exact same type 14 a few years down the line.
So to make a truly effective cold vaccine, you'd need to formulate a shot with 150 different active components.
Which is just technically impractical right now. We don't have the pharmaceutical technology to make a single cocktail that broad, and the virus mutates too fast anyway.
I want to use the text to bust a major myth here. My grandmother, and probably everyone's grandmother, always said, don't go outside with wet hair, you'll catch a cold, or put on a coat, or you'll get sick.
The source material is very explicit about this, actually. Experiments under highly controlled conditions have shown that chilling, including specifically wearing wet clothes in the cold, does not produce a cold or increase your susceptibility to the virus.
So the physical cold weather does not make you sick.
No. But cold weather drives people indoors. It leads to crowding, poor ventilation. We close the windows, we turn on the heat, and we sit close to each other. And that proximity is how the virus spreads.
Hand to hand, hand to object.
Exactly. These viruses are incredibly hardy on environmental surfaces. Remember that beta barrel armor shell we talked about at the beginning?
The tank.
Right. It allows them to survive for hours on a doorknob, a keyboard, a shared pen. You touch the infected knob, and then you absentmindedly rub your eye or scratch your nose. Bam! Self-inoculation.
So the treatment is just deal with it.
Symptomatic relief. Drink fluids, get some rest, take something for the fever. That's really it.
Let's pivot to the final major group, the outlier of the family. Foot and mouth disease.
The aphthovirus genus. Now, we need to be very clear right up front. This is not the same as the hand, foot, and mouth disease in humans that we discussed earlier.
Hand, foot, and mouth is human, caused by Coxsackie.
Yep.
Foot and mouth is aphthovirus, and it affects animals.
Correct. Foot and mouth disease targets cloven-hoofed animals, cattle, sheep, pigs, goats. It is notoriously, devastatingly contagious among livestock.
Why is it such a massive big deal globally if it doesn't really hurt humans?
Pure economics and food security. It causes painful vesicles in the mouth and on the feet of the animals. Because it hurts to eat and walk, they rapidly lose weight, and dairy cows stop producing milk entirely. It absolutely devastates agricultural industries.
And the control measures described in the text are pretty extreme.
Quarantine and slaughter. If an outbreak is detected, entire herds, even healthy animals nearby, are destroyed to stop the spread. You might remember those horrific news images from the UK in 2001, just massive burning pyres of cattle and sheep.
Yeah.
It's not because the virus kills them all, many animals would survive, but because the only way to save the broader industry and stop the virus from becoming endemic is to completely destroy the host reservoir. Humans are very rarely infected. It's strictly a massive animal health and economic crisis.
Finally, let's zoom out a bit and look at how these viruses move through the broader world. The environmental transmission section in Chapter 36 has a diagram, figure 36-4. That is honestly a bit gross to think about.
It is the circle of life, but specifically for enteric viruses. It maps the flow from human excreta to raw sewage to the municipal water supply into the environment and back to humans.
The key point it hammers home is durability. These viruses are incredibly tough outside a host.
They are. Enteroviruses in particular routinely survive standard sewage treatment and standard chlorination levels far better than many common bacteria do. They are non-enveloped viruses, which makes them highly robust against detergents and environmental stress.
And there's a specific warning in there about shellfish.
Yes, oysters, mussels, and clams. They are filter feeders. They sit in the water and constantly pump gallons of it through their system to extract microscopic food. If that coastal water is contaminated with human sewage runoff...
The shellfish filter out and concentrate the virus directly in their own tissues.
Exactly. And because we often eat oysters raw on the half shell, we bypass any heat that would kill the virus. It's a very efficient, direct transmission vector from sewage back to the human gut.
So eating raw oysters is essentially playing a mild game of Russian roulette with enteroviruses if the water source isn't pristine.
It's a known risk factor, yes. Cooking them thoroughly eliminates the risk, but raw is a different story.
So, man, we've covered a lot of ground today. We've gone from the tiny 30-nanometer canyon structure on a geometric shell to the global eradication campaign for polio to the sewage cycle on oysters. Let's try to synthesize this for the listener. What is the main takeaway here?
For me, it always comes back to the terrifying efficiency of the Picornavirus design. It uses such a microscopic amount of genetic code, just a few thousand RNA bases, to build a machine that can survive stomach acid, hide its weak points from our antibodies using that canyon structure, completely hijack complex human cells, and spread through multiple environmental routes.
It is biological minimalism at its absolute finest, or its absolute worst, depending on how you look at it.
Exactly. It uses the exact same basic toolkit, the beta barrel, the hidden canyon, the polyprotein cleavage, the RNA genome, to attack the nervous system, the heart muscle, the skin, and the respiratory tract. It's remarkably versatile.
It is a sobering reminder of the microscopic arms race we are constantly in. You know, we are winning against wild polio, which is amazing, but the family has so many other members. The canyon structure alone is a testament to how evolution continually finds a way around our best natural defenses.
It's a perpetual stalemate. We have engineered incredible vaccines for some, but we are completely vulnerable to others.
So what's a final thought we can leave the listener with?
I'd say this. We often spend our time fearing the exotic, dramatic viruses, Ebola, Zika, things like that. But the Picornaviruses are the ones silently living among us every day. On our doorknobs, in our daycares, in our water. The fact that polio is nearly gone is a triumph of modern science, but the sudden emergence of paralytic EVD68 shows us that the neurological niche for these viruses is still wide open. They're always probing our defenses.
They aren't going anywhere. And it really makes you wonder, given the diabetes connection with Coxsackie-B, what other chronic lifelong conditions are secretly just the aftermath of a picornavirus we caught when we were 5 and forgot about?
That is the million-dollar question in virology right now.
Well, on a more practical note, what can people actually do?
Wash your hands. Seriously, it sounds basic, but it is the only reliable defense against a non-enveloped virus that can survive acid and live on a table for hours. Hand sanitizer often isn't enough for these guys. Good old-fashioned soap and water physically removes them from your skin.
Simple advice for a highly complex enemy. Well, that's a wrap on Picornaviruses. Thank you for taking this microscopic plunge with us today.
It was a pleasure to be here.
We see you all on the next deep dive. Keep learning.